Hi, everyone. Welcome to the Society for Developmental Biology's first annual postdoctoral seminar series. We're excited to highlight the work of our outstanding postdoctoral members. Today, Dr. Melissa LaBonty from the University of Alabama at Birmingham and Dr. Isabella from the Fred Hutchinson Cancer Research Center will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. Our first speaker today this afternoon is Dr. Melissa LaBonty. Melissa is an NIH IRA CDA Merit Postdoctoral Fellow in Dr. Bradley Yoder's lab at the University of Alabama at Birmingham. It is also adjunct faculty at the University of Montevallo. She did her PhD in cell, molecular, and developmental biology at Tufts University with Dr. Pamela Yellick, studying skeletal diseases using zebrafish. In addition to her productive research career, she's highly involved in university teaching and mentoring, including having mentored 24 undergrad students and 2 graduate students. So today we'll hear from her about her postdoctoral work studying the conservation of the genetic interaction between BBS5 and MPHP4 across species. So please join me in welcoming Melissa.
Thank you, Nicole, for that great introduction. And thank you to the organizers for allowing me to come and talk to you today about my work as a postdoctoral fellow in Brad Yoder's lab. So today I'm going to be talking a little bit about the conservation of the genetic interaction between BBS5 and MPHP4 across species. And for today's talk, I'm going to start by going into a little bit about primary cilia and why they're important. Then I'll move on to talk about why we use C. elegans to study the primary cilia. I'll go into our project looking at the genetic interaction between BBS5 and MPHP4 in C. elegans. Then I'll move on to talk about how we've observed the conservation of this interaction in other organisms. And finally, I'll address some of our future project directions. So I want to begin by actually bringing in one of my favorite analogies. I like to think of the primary cilia as kind of like an air traffic control tower. So when you think of one of these buildings, you know it's a space where a lot of critical messages are sent and received, and that's happening constantly to ensure that airplanes are going to the right place at the right time. Now, imagine if cells had one of these air traffic control towers. Maybe they'd have a way to respond to cues from the surrounding environment or to send out their own signals in order to influence cellular behaviors. So as it turns out, cells do have their own signaling hub of sorts, and it's called the primary cilia. So primary cilia look kind of like this is a pseudo-colored picture here, but they're solitary specialized compartments found on nearly every mammalian cell type. So what do primary cilia do? Well, they're capable of detecting physical forces that are exerted on cells. They can sense chemical changes in the environment outside the cell, and they send and receive signals between cells. And that's just a small number of the things they're responsible for. So how do we build a primary cilium? Well, you start with a foundation at the base, and that's called the basal bodies. And from there, you can assemble a microtubule-based tower called the axoneme. Then you surround that strong tower with a lipid bilayer to create a compartment. Then to ensure that whatever gets in or out of that compartment is actually what you want, you add a selective barrier called the transition zone. And then you need things that can actually transport across this transition zone. And these are typically thought of as intraflagellar transport complexes. You can also have a complex called the bibisome. And finally, you need to bring cargo into the primary cilia by those transport protein complexes. And that's going to include an array of receptors that are responsible for sensing and signaling. So let's go back to that idea of the air traffic control tower. And now I want you to imagine what would happen if the people working in the air traffic control team were not there anymore. You'd probably imagine there would be delays in air traffic, you'd have incorrect routing, and all around, it wouldn't be a great situation. So when cells lose their primary cilia, something similar happens. Basically, what it leads to is a family of disorders called ciliopathies. And ciliopathies are characterized by a wide array of phenotypes including cleft palate, cognitive defects, liver disease, renal cysts, retinopathy, and even polydactyly. So you can see there's really a lot of systems that are affected by the loss of cilia function. And many ciliopathies actually present with a lot of highly variable clinical features. So it's important to think about the fact that patients with mutations in ciliary genes don't always present with all of the features that I've mentioned there. For example, the ciliopathy nephronophthisis is associated with mutations in the NPHP family of genes, which is a known ciliary family of genes. And you can see from this figure that there's a huge array of clinical features that are associated with this disease, but if only a few of these phenotypes are actually prevalent in more than 20% of cases. So sometimes patients with the same known mutations can present with more or less severe clinical features. And then this brings us to our working hypothesis right now, which is basically that we think that there's this phenotypic variability that's stemming from the presence of multiple variants that are causing different degrees of primary cilia dysfunction. So we show this in our model here, where we think that a single gene mutation might cause a small decrease in ciliary dysfunction that could be exacerbated if there's a secondary mutation that causes a greater decrease in ciliary dysfunction when compared to the single variant. So this kind of hypothesis is really difficult to study in human patients, as you can imagine. Instead, it's a lot easier to be able to study this sort of thing using a model organism, especially my favorite model organism, C. elegans, which is this wonderful, small, fast-growing, easily manipulated organism you can see in the video here. So one of the advantages of using this system is that C. elegans only have primary cilia on their sensory neurons instead of on all of the cells throughout their body. So the sensory neurons are found in the head and the tail of the animals. The sensory neurons in the head are referred to as amphid neurons, and the sensory neurons found in the tail are referred to as phasmid neurons. And when we disrupt the function of these sensory neurons, the animals can still grow and reproduce. And that's really important because that means we can still study what's going on when primary cilia are not functional. What happens instead is that sensory mediated behaviors are disrupted. And so we can actually study those sensory mediated behaviors in a number of different ways. So this is just a subset of some of the ciliated sensory neurons that you can see here. And we can study some of these different subsets based on the kinds of behaviors that they modulate. For example, these sets of neurons are responsible for chemotaxing behaviors. These neurons are directly exposed to the external environment, so they can take up fluorescent lipophilic dyes. And finally, this set of neurons here is responsible for detecting physical touch. So there's a wide array of different kinds of assays that we can use to be able to understand what's going on with ciliated sensory neurons. So in our lab, we use dye filling as a primary tool to identify proteins involved in ciliary function. And we think that this is a great tool because you can use it very quickly to identify things that are involved in ciliary function. So for example, all we need to do is swim animals in this dye, and if they have normal primary cilia, they'll be able to take up these lipophilic dyes and they'll fill the amphid and phasmid neurons, or at least the ones that are exposed to the external environment. And this dye doesn't just fill the primary cilia, it also fills the dendrite, the cell body, and the axon of these neurons. In contrast, when animals have defects in primary cilia formation or function, we'll see one of two things. Either the dye has a spotty or partial dye filling pattern, or there's no dye at all in the neurons. And so we can use this as a readout to identify when primary cilia function is lost. So what we did is we went back to this knowledge of the variability of clinical features in nephronophthisis patients, and we looked at what genes involved in nephronophthisis are conserved in C. elegans, and we saw that NPHP4 is one gene that is well conserved. And we thought it was a really interesting candidate to follow up on, especially because animals that just have a loss of function in NPHP4 don't actually display dye-filling defects on their own. And not only have we demonstrated this result, but other labs, including the Reiter and LaRue labs, have shown this as well. So what is NPHP4? Well, it's one of at least 20 proteins that work together in the transition zone to establish that gating or barrier function of the primary cilia. So we employed forward genetics to be able to identify genes that might be working in tandem with the mutation in NPHP4 to cause a dye-filling defect. And when we did this EMS mutagenesis screen, we were able to isolate 29 different strains from the screen, including one that carried a mutation in the gene BBS5. So what is BBS5? Well, BBS5 is, fortunately for us, also a known ciliary protein. It's part of this 8-member complex called the BBsome. And the BBsome is known to work in concert with the intraflagellar transport machinery to transport membrane cargoes into and out of the cilia. So we were excited to see that this genetic interaction between BBS5 and NPHP4 has been previously demonstrated using dye filling, which is great for us because it provides additional rationale that we should be pursuing this to further characterize the interaction. So here's what we were able to find when we looked at dye-filling defects in our lab. And I want to point out here, we didn't just look at the dye-filling in the amphid neurons, we also looked at dye-filling in the phasmid neurons. And another thing we wanted to do, we isolated a BBS5 allele in our lab, the YHW62 allele. We also wanted to compare that allele to a previously identified allele, the GK537 allele. And we saw a few interesting things when we did these analyses. So the first thing we saw, which is is that, of course, there is a loss of normal dye filling in a majority of the animals harboring both the NPHP4 mutation and either BBS5 mutation. So you can see that the dye filling is essentially almost completely lost in the mutation that we isolated from our screen, and it is partially lost in the mutation that existed previously. We also found another surprising result, which is that the BBS5 allele isolated specifically from our screen causes dye-filling defects in the phasmid neurons, but this defect is not phenocopied in the existing BBS5 allele. So together we realized all of this data really does suggest that mutations in NPHP4 and BBS5 are working together to enhance ciliary dysfunction. So we've gone on to characterize this genetic interaction to determine whether there's conservation in other model systems and also to learn more about the mechanism behind the interaction. And this has been a group effort between myself, graduate student Melissa Bentley Ford, our current lab technician Cameron Lafayette, and an undergraduate Michaela Scott. So we were interested in coming up with a number of assays that could help us to determine how the primary cilia are actually affected by this genetic interaction. So we've taken 2 tracks for this. The first is to look at behaviors that are connected to sensory neuron function, and these include looking at things like chemosensation or dower formation or egg laying. And the second is to use reporters to actually analyze the ciliated sensory neuron structure as well as its function. And to do this, we've taken advantage of transition zone integrity markers, receptor localization, and also looked at intraflagellar transport. And so I'm just going to show you a couple of the results that we've gotten so far. We've looked at some of the behavioral assays, and what's really interesting is that we actually get contrasting results depending on the assay that we use. So when we look at chemosensation, we can see that the NPHP4 and BBS5 single mutant alleles cause essentially almost a complete loss of the ability to chemosense. And because they are causing this loss of chemosensing ability, we really don't see an additive effect when both mutations are lost at the same time. And this goes for a number of different attractive compounds that we looked at across the spectrum. On the other hand, when we look at Dower formation, what we see is in contrast to what we found with chemosensation, where single mutants alone don't have defects in forming Dowers. But when we have mutations in both genes, then we do see defects in Dower formation. Also, looking at fluorescent reporters, what we're able to see is kind of similar to what we saw with the chemotaxis assays. So this reporter is a marker for transition zone integrity. And in a wild type animal, this reporter stays outside or is excluded from the primary cilia, which is marked by this blue bar here. In any of our mutants, either animals carrying just a single mutation or in the double mutant lines, we can see that the transition zone integrity is lost, and this reporter is able to essentially leak into the primary cilia. So what do we actually think is the mechanism driving this interaction? Well, it's kind of up in the air right now. We have two different disparate kinds of results. We have results like dye filling and the Dower formation assay that do suggest that there's a genetic interaction. But we also have results that we can't really make a strong conclusion from or don't necessarily suggest a genetic interaction, like the chemotaxis assays or looking at transition zone integrity. So what we want to do to follow up on this is really to look at what is shared between these, the dye filling assay and the Dower formation assay, to try to identify a mechanism that might be driving this interaction. And ultimately, we're hoping that this interaction can explain some of the variability we see in human ciliopathy patients. So to follow up on this, we're also using two different models, zebrafish and mice, to find out if this interaction is conserved in vertebrates in order to justify whether this interaction is actually conserved in other mammalian species. So we also have kind of a contrasting picture when we look at these two vertebrate species. When we look at zebrafish, BBS5 and NPHP4 in this animal don't actually appear to display any obvious genetic interactions. So what we see here by micro CT is that BBS5 single mutant zebrafish have scoliotic defects that are essentially phenocopied in double mutant animals. And if we go on to do histology and look at tissues inside the animals, we can see that vertebrae, kidneys, and the heart all essentially look identical and unaffected in these animals. We also went on to look at the eye, and in the eye, we see similar phenotypes to what occurs with scoliosis, where there's defects in the outer photoreceptor layer in the BBS5 single mutants that look nearly identical in the double mutants. So let's head back over to the mice. And when we're looking in the mice, again, we see a different picture. So BBS5 animals, when we look at congenital mice, are actually not detected at weaning age. So what we're seeing here is that BBS5 animals, single mutant animals, are viable at sub-Mendelian ratios, but we can see them. And BBS5 NPHP4 double mutant animals are never observed. And so we went to look at embryos before weaning age. So we collected embryos at day 16.5. And what we can see is that they actually look morphologically normal. So by contrast enhanced micro CT, we can't actually detect any obvious defects that would explain the loss of these animals at weaning age. So this is something that we're interested in following up on. And we've also done juvenile induction of loss of BBS5 in an NPHP4 background. And we see something similar here that also suggests there's a genetic interaction. Using this conditional BBS5, we can see that when they're induced at 7 days post-fertilization, we get ubiquitous loss of BBS5, and we also end up seeing an abrupt death in most of these animals. And another thing that we see in these animals before they die is that the juvenile-induced double mutants develop seizure-like behaviors. We aren't yet sure what the cause of this phenotype actually is, but it could point to some neurological defects in these animals. Importantly, we do know that when animals are induced as adults, they don't develop this problem and they don't end up dying abruptly.
Hey, Melissa, just 1 minute left.
Okay, thank you. I'm just going to wrap up. So here's what we have concluded from our project so far. We know that there is a genetic interaction between BBS5 and NPHP4, but it appears to have very specific effects in C. elegans. So it does cause dye filling and dower formation defects, but we don't see any difference when we look at chemotaxis or at transition zone integrity. We also noticed that there isn't really a genetic interaction between BBS5 and NPHP4 in the zebrafish. And finally, we were able to recapitulate this genetic interaction between BBS5 and NPHP4 in mice. And we're wondering if maybe this interaction might be tissue specific. So where do we see this project going? Well, first off, we definitely want to use C. elegans to be able to try to identify this mechanism that's driving the genetic interaction between BBS5 and NPHP4. And we think that looking at dye filling and dower formation and seeing what things overlap between those two is really going to be key here. We're also interested in studying the different BBS5 alleles that we have in C. elegans to determine why they can cause different phenotypes. And finally, we want to know if this genetic interaction is specific to NPHP4 in the transition zone and BBS5 in the BBsome, or if there's more components within these complexes that might also be able to genetically interact. And so with that, I'd like to thank the lab. We've had a lot of people working on this awesome project. And in particular, I'd like to thank our funding sources. So I'd love to take any questions from the audience now.
Okay, that was a really great talk. So just a reminder to the audience, you can type in your questions in the Q&A box down at the bottom of your Zoom screen. So we have some questions coming in already. Okay. Brandon Carpenter is asking, you may have gotten to this, but what are the mutations in the two different BBS5 alleles? Are they in similar domains?
Great question. So the BBS5 allele that we identified from our EMS mutagenesis screen is a single point mutation that introduces an early stop codon. And that stop codon is found in exon 3 of the gene, and it would potentially result in a fairly highly truncated protein, but as far as we can tell, no product is actually made. The second allele, the GK537 allele, is a large deletion that actually takes out the entire promoter and the first two exons of BBS5 in worms. We don't really know right now why it is that such a deletion wouldn't actually result in the same phenotypes as this early stop codon mutation that we've identified. But we're interested in following up to see if there may be some alternatively spliced isoforms in the deletion allele that allow some sort of functional product to still be made.
All right, Emily Bayer is asking, is it possible that a redundant copy is rescuing the mutant in fish due to the genome duplication?
That's a really good question. So we have not had the chance to do an extensive look at the zebrafish genome yet to really determine whether that is the case. But having worked as a grad student in zebrafish for a while, I did know about the genome duplication. So I think that's a really great avenue for us to check to make sure that we've truly knocked down BBS5 function completely.
All right. Tamara Caspary is asking, so two questions. First, do you know whether either C. elegans BBS5 alleles assemble in the BBsome? And the second question, do you know whether what you're calling leaky is due to a problem with ciliary entry or exit?
Okay. So first question, do we know if either of the alleles are found in the BBsome? At this point, we have not done the biochemical analysis to determine whether the transcripts are actually made, anything is made into a protein and therefore found in the BBsome. And to add on to that, we haven't had the chance to look yet to see if the BBsome itself is still being made and going into the cilia like normal without the presence of BBS5. So these are definitely things that we want to continue to follow up on. Can you remind me what the second question was?
Yeah, the second question is, do you know whether what you're calling leaky is due to a problem with ciliary entry or exit?
Okay. So essentially, the RPI2 protein is something that is supposed to be maintained in the periciliary region of the cilia. And if something goes wrong and it's able to get into the cilia, I believe that it's a leakiness of it being able to get transported or even just diffuse into the primary cilia because the transition zone is no longer able to maintain it outside of the cilia.
All right, and then Clarissa Coveney is asking, hi Melissa, great talk. Just checking the mice are whole body deletions and
Yes.
the skeletal phenotypes are very interesting. Do you know if there are any matrix changes that can occur pre-ossification? Because sometimes musculoskeletal phenotypes can recover later.
So I'm guessing the musculoskeletal phenotype is in reference to the scoliosis.
Probably, yeah.
Okay. So what we do know about the scoliosis that is occurring in the zebrafish, this is actually a fairly common phenotype that's seen in a lot of ciliary mutants. So we weren't surprised to see that phenotype occur. And at this point, I think most researchers who work on that more extensively than we do have found that that seems to be an effect of the Reisner fibers not working properly and establishing the right flow so that the spinal cord forms properly and then the vertebrae form properly as well. So we haven't followed up on that extensively. We'd love to be able to get a reporter from somebody to be able to look at whether the Reisner fibers are also disrupted in our BBS5 mutants. I'm sure that they would be, but we haven't looked at that at this point. Was there, I think there was a first part to that question maybe.
Oh yeah, you answered they were whole body deletions, the mice.
Okay,
right?
yeah.
Okay, great. And then we have time for one last question. So this one's from Kara Morovich. Great talk. Do you see any sperm defects in any of these models?
Good question. The answer to that is yes. And I believe, I'm not entirely positive, but I believe that that exists in a publication that just came out of our lab from Melissa Bentley Ford on just looking at BBS5 single mutant mice. So if you want to go and take a look at that, definitely check out her recent paper. I think it just got accepted a couple weeks ago. So that would be a great one to read.
All right, great. Thank you, Melissa. We'll move on to the second speaker now.
Okay, great. Thank you, Melissa and Nicole. That was really nice. Now I have the pleasure of introducing Adam Isabella, who did his PhD at the University of Chicago, where he studied how basement membrane remodeling influences epithelial morphogenesis in Drosophila. And now he is a postdoctoral fellow with Cecilia Moens at the Fred Hutchinson Cancer Center in Seattle, where he's been focusing on the development and regeneration of the nervous system in zebrafish, which he's going to tell us a little bit about today. Adam, go ahead.
Okay, thank you. So it's great to be here. Thanks, Chase, for the introduction, and thanks everybody for coming. I'm really excited to be a part of this series. In the Moens lab, we're really interested in understanding how the nervous system is organized. So this image that we're looking at here is one of the larval zebrafish head, and in green are labeled the branchiomotor nerves. And these nerves are collectively responsible for innervating and controlling the majority of muscles in the head and neck. And in order for them to do that properly, there has to be a really precise developmental process in which the neurons of these nerves have to aggregate in the correct places in the brain, and then they have to extend axons over long distances along very specific paths in order to innervate the appropriate tissues. And so we're really interested in understanding what are these developmental mechanisms that allow all of these axons to get to the right places and ultimately build this complex branched structure to control muscle function. Now, we've been specifically focusing on the vagus nerve in this context. So this is the most posterior of these nerves. It's composed of this large cluster, also called a nucleus of neurons in the hindbrain. And each of these neurons sends out an axon, and these axons all travel along a common path out of the brain until they reach this choice point here in the periphery. And at this point, each axon has to make a decision to go to one of 5 distinct branches. So these branches innervate a number of tissues. They innervate the posterior pharyngeal arches. So they're named branches 4, 5, 6, and 7 for the arches they innervate. And there's also this one more posterior branch called the visceral branch that goes back and innervates a bunch of visceral organs. And this branch structure is actually what got us really interested in the vagus nerve. Because if you look at the functions of these branches, they're actually quite diverse. These more anterior branches control things like swallowing and coughing, and then more posteriorly, you see control of things like heart rate and digestion. And it's probably easy to imagine that the control of all these different processes has to really be independent, such that each one is controlled at the right time and place. And so we're really curious to understand how could it be that this single group of neurons is organized such that it can control all these different functions at the right time and place. And so I want to introduce to you a common organizational principle in the nervous system that's really important for the purpose of this talk, and that's what's called a topographic map. So topographic maps are situations in which the positions of neurons in the neural tissue are indicative of the positions that their axons are going to innervate in the target tissue. And so probably the best example of that is in the retina, where you've got neurons positioned at different points along the dorsal ventral axis of the retina. And based on their position along this axis, they innervate distinct regions of the tectum along the medial lateral axis. So more ventral neurons innervate more medial positions, more dorsal neurons innervate more lateral positions. So you see this commonly in sensory systems as well as motor systems. Another really nice example is in the innervation of limb muscles, where spatially distinct pools of neurons in the spinal cord innervate distinct muscles. And so we're really curious to understand what is the sort of analogous organization of the vagus nerve. And to address this question, I came up with a method to really carefully map the innervation of this nerve using a single cell transgenic labeling approach. So you can see by labeling this single cell in green, I can really carefully measure its position within the nucleus. I can also measure its innervation target. And by doing this for neurons all over the nerve, I was able to build up this map, which is represented as a position probability matrix, where for a neuron at any given position along the anterior posterior axis of the nerve, we know the probability of innervating each branch. And you can see a real clear topography that comes out of this, where the most anterior neurons here on the left are innervating the most anterior branches, 4 and 5. And as we go progressively more posterior in the nucleus, you see progressive innervation of more posterior branches. And so that is illustrated here on the right, where we now think of this nerve as being sort of subdivided spatially such that there are these distinct target groups, which are color-coded, of neurons here that are responsible for innervating distinct branches, and that we think this spatial separation may play an important role in kind of allowing for somewhat independent functions of these groups. But that brings up the really interesting developmental question of how does each of these neurons know precisely where to extend its axon during development in order to build up this pattern? So one of the things that we learned really early on in studying this was that these neurons are interpreting environmental signals to tell them whether they're anterior or posterior neurons. And we were able to know this through a transplantation experiment where we can actually take neurons out of one embryo and we can stick them back into the vagus nucleus of another embryo. And so if you take, say, an anterior neuron and you transplant it into the posterior of another animal, you can ask, does it retain a knowledge that it's an anterior neuron and target appropriately, or does it change its identity to posterior now based on its new environment? And the answer is that it very clearly changes its identity and now targets posterior neurons, which means that it's interpreting information in the environment to tell it now it's in a posterior position. So this is an illustration of that. Here's the actual data. We've got these few transplanted neurons that were anterior, but now are targeting to posterior branches. So the next thing we wanted to know was, what are the molecular differences between anterior and posterior neurons that might be allowing them to make different targeting decisions? So to do that, we decided to take an RNA-seq approach, where we expressed a photoconvertible protein in these neurons. We then photoconverted either just anterior or just posterior populations. And then we isolated these cells and we did RNA-seq. And so what we're looking at here are genes that are enriched in the anterior cells and genes whose expression is enriched in the posterior cells. And what this experiment ultimately showed us was what that environmental regulatory signal was that was telling neurons if they were anterior or posterior. And the hint to that was that a lot of these genes that we found enriched in the posterior neurons were known to be responsive to retinoic acid signaling. So retinoic acid is a pretty well-known morphogen that mediates certain aspects of anterior-posterior patterning. And sure enough, we saw that if you look at a retinoic acid reporter, along with these neurons at the time that they're extending axons, you can see, in fact, that retinoic acid is at pretty high levels over the posterior neurons, at pretty low levels over the anterior neurons. And so it seems like retinoic acid is in the right time and place to be telling these neurons who they are. But what we really wanted to know next was, is it actually informing these neurons about where to extend their axons? And so to address this question, we did sort of a variant of the single cell labeling approach. So this is a control animal here. It's the same one I showed you earlier, where I'm just expressing GFP in this cell. And this is the control map that we get from that. The twist that I put on this experiment is that in addition to expressing GFP in this cell now, I also express a dominant negative retinoic acid receptor. And so that's going to drive down retinoic acid signaling in these green cells. And this has a really dramatic effect on the map, where now even the most posterior neurons, when you turn down retinoic acid, are extending their axons to aberrantly anterior branches. We can do the same thing with the constitutively active retinoic acid receptor. And we see the opposite thing, where now even very anterior neurons, if you turn up retinoic acid, are going to posterior branches. And so it seems really clear that each neuron is making a decision individually based on how much retinoic acid it's seeing. If it's getting a high level of retinoic acid, it goes posterior. If it's getting a low level, it goes anterior. So that was really informative. But the next thing that we wanted to test was, what are the genes that are being controlled downstream of retinoic acid that are actually mediating these targeting decisions? And so to understand that, we again turned to RNA sequencing. This time what I did is I took whole animals as they were developing and I bathed them in retinoic acid to turn it up in all of the neurons. And then I isolated vagus neurons and I sequenced them. And so this graph is showing all the genes that are downregulated as a result of retinoic acid treatment and all the genes that are upregulated as a result of retinoic acid treatment. And a lot came out of this, but the one that I want to focus on today is just this one gene that's in the downregulated category called MET. And the reason MET is what we've decided to focus on out of the screen is that this is a axonal receptor that's been implicated in other systems in axon guidance. And so we thought it might be a good candidate to be controlling axon guidance in this system. So we can validate these results by RNA in situ. So if I look at MET RNA, which is in purple here, you can see that it is expressed in the vagus neurons in the low retinoic acid domain. And importantly, its ligand, HGF, is expressed in the pharyngeal arches. We're actually intrigued to see that actually both of these genes are being negatively regulated by retinoic acid. So if you treat animals with DEAB, which is an inhibitor of retinoic acid synthesis, you see that the MET expression expands to fill the entire vagus domain. And HGF expression also expands to fill the entire pharyngeal domain. So retinoic acid seems to be suppressing them both. We also showed that MET and HGF are required for the targeting of these axons. So we generated mutants for these genes, and they both show the same phenotype, where in control, normally we see this nice innervation of the arches, as I've shown you previously. In a MET mutant or an HGF mutant, the exons come down to the choice point. They have no trouble getting it that far, but then they stall out. And they really have a lot of trouble actually finding their way into the arches. So it seems like HGF is drawing these axons that express MET into the arches. So that was very informative. We know that MET and HGF are involved, but you may notice that they're required for innervation of all of the branches. And so this didn't really give us a sense of how MET HGF signaling might be driving differential targeting based on a neuron's position. And so to understand that, I need to introduce you to another sort of quirky aspect of the development of this nerve. I'm going to show you a movie here, because what we saw is that there's this interesting temporal patterning of innervation. So what you're going to see is these axons coming down and innervating the branches, and you're going to see an anterior posterior sequence of innervation, where the first axons to come down all initially innervate only PA4. And it's only several hours later that there's a switch that happens and PA5 starts to be innervated. And it's only if we wait another several hours, like 6 to 8 hours later, that we see a switch and then PA6 starts to be innervated. So it seems like there are these distinct windows in time when each arch is innervated, and that progresses from anterior to posterior. And so because of these temporal dynamics, we thought it would be important to look at the temporal dynamics of MET HGF signaling and see if that gave us a clue as to how they might be controlling targeting. And indeed, we did see a really dynamic expression of both of these genes. And in both cases, what we see is this wave of expression from anterior to posterior that sort of sweeps through these tissues during the development of this nerve. So for MET, if you look at an early stage, you can see it's really only expressed in these anterior-most cells. And over time, it moves posterior such that at sort of a middle time point of development, about half the cells express MET. And at a late time point, they've pretty much all turned it on. And something similar happens with HGF, where at an early time point, it's really only expressed in this anterior-most arch, arch 4, and then it progressively turns on in more posterior arches, first 5, and then 6. And so this information, sort of coupled with what we knew previously, gave us what we thought was a really clear model for how this might be working. And so what I'm going to do is I'm going to present the model, and then I'm going to give you a little bit of validating data for it. So what we think is happening, and we call this the temporal matching model, is that at an early time point in development of this nerve, retinoic acid is coming from the posterior, and it's repressing MET expression in all but the anterior-most of these neurons. And it's also repressing HGF expression in all but the anterior-most arch. So that means only these neurons are competent to innervate, and only this arch is competent to receive them. And so they become matched. And then what happens over time is that retinoic acid starts to recede towards the posterior. Now what that does is that it de-represses MET in a new group of neurons at the same time that it de-represses HGF in a new arch. So at the time that this arch turns on and starts drawing axons in, these are the neurons that have become competent to innervate, and those two groups become matched. And that just kind of happens down the line as time proceeds. Retinoic acid goes back, MET turns on, HGF turns on in a more posterior arch, and those two groups become matched. And that ultimately drives that spatial patterning that we see. Okay, so now for the validation of this. We were excited about this model because one of the things that we thought was a clear hypothesis that came out of it was that if we take these anterior neurons and we delay when they're able to turn on MET signaling, that's going to cause them to miss their opportunity in time to innervate anterior arches, and it's going to drive them posterior. And we thought the longer that we delay them in their ability to signal through MET, the more posterior they're going to go. And we can do that using this drug crizotinib, which is a small molecule MET inhibitor. So you can see that if I treat animals with crizotinib, I get a really nice phenocopy of the MET mutant where these exons stall. But the nice thing about this is that I can wash the drug out anytime I want, and I can rapidly reestablish MET signaling. And so what I did for this experiment is I did 2 pulses of crizotinib, sort of a short pulse and a long pulse to try and get a more severe effect. And then after I washed the drug out, I allowed axon targeting to continue. And then once it was over, I asked what happened to these anterior-most neurons by photoconverting them. And it's exactly what I expected. So if we don't treat with crizotinib, you can see that these anterior neurons predominantly go to arch 4. If we do a short pulse, they fail to innervate arch 4, and they instead go to arch 5. And if you do a long pulse, you see a more severe phenotype where they fail to innervate arches 4 and 5, and they instead go to 6. And so that suggests that they missed the opportunity in time when I had turned off MET to innervate these anterior structures, and when I turned MET back on, PA6 was the one that was being innervated, and so that's where they went. Okay, And so to conclude this first part of the talk, what we've done is I think we've sort of added a new dimension to the understanding of how topographic maps develop. This had previously been thought of as largely a purely spatially regulated targeting system. In this case, we've shown that in the vagus nerve, not only is there important spatial information at play, but there's really careful coordination in the timing of signaling between neurons and their targets that allows these axons to make really precise and complex targeting decisions and ultimately build up this complex map that we think is important for the multifunctionality of this nerve. All right, so for the last 5 minutes, I want to switch gears a little bit and talk about some more recent work that we've been doing related to the regeneration of this nerve. And there are a couple of reasons that I got excited about regeneration in this system. First, I would say that I think axon regeneration is kind of the second time in a neuron's life when it might find itself having to extend an axon. So if there is, after this nerve has developed, some insult or injury that breaks those axons, they're going to lose their ability to communicate with the tissue. And in order for them to reestablish that, they're going to have to regrow, and they're going to have to do that in the correct pattern again in order to reestablish proper function. The second reason I got excited about regeneration was based on my thinking about how development was going on. Because sort of the whole point of the first part of this talk was that it seems like everyone has to be doing their thing at exactly the right time in order to build this right. These guys have to go out early, these ones late. And there's really no way that that sort of temporal component could be recapitulated during regeneration, because sort of by definition, regeneration is happening late, after embryogenesis has happened. So for instance, those MET and HGF waves have swept through the animal way back in time, and these regenerating axons are really not going to be able to use that temporal matching model in order to get where they're going. So we thought if they're able to regenerate, there's going to have to be some new mechanism that might be really interesting to find out about. And so let me introduce you to the method that we're using to study regeneration, which is a single neuron transplantation approach. So I mentioned to you earlier in the talk that we can transplant neurons between animals in the embryo. We found that we can also do that in the larva. And so if I take a neuron from one animal and pluck it out at the larval stage after it's already formed its initial axon, I can put it back in another animal, and this does a couple of things for me. The first thing that it does is it tears the axon away from the cell body. And so that's going to force that cell to regrow an axon in its new environment. It also allows me to really easily track this cell because it's a different color than all the other cells. And the third thing it does, which I'll show you in the next few slides, is it allows us to really precisely and in interesting ways manipulate this cell experimentally. So it turns out that this approach works really well. We have really high success rates where this is an example of a transplanted neuron. If we give it a couple of days, it'll happily regrow an axon, and that axon will come down and target one of these branches. And the first question we wanted to ask, of course, is this appropriately patterned? So for instance, if we take an anterior neuron and we transplant it back into an anterior position, can it reliably regrow to an anterior branch, or does it get confused and just go all over the place? And the answer is that it reliably regrows to the right place. So this is that image I just showed you. This is actually an example of an anterior to anterior transplant where we see a nice return to the anterior branch. And if we quantify that over many animals, you can see that in anterior to anterior transplants, you really only see regrowth to the anterior branches, 4, 5, and 6. And if we do posterior to posterior transplants, you really only see regrowth to posterior branches, 7 and visceral. The next thing that we wanted to know, similar to what we asked in development, was, does this neuron have an intrinsic knowledge of its position, anterior or posterior, or is it interpreting environmental signals? So again, we did this anterior to posterior transplant. And then you can ask, does this cell still think it's an anterior neuron and go to anterior position? Or does it now change and become a posterior neuron and go to a posterior position? And what we saw here was the opposite of what we saw in development. In this case, this cell really remembers where it came from. So this is an example of an anterior to posterior transplanted neuron. And you can see that it's regrown its axon still back to the anterior. And this holds up pretty well across many animals. In an anterior to posterior transplant, you still predominantly see anterior targeting. In a posterior to anterior transplant, although it's a little bit messier, you still predominantly see posterior targeting. So it seems like in this case, these neurons really know who they are, and wherever you put them, it doesn't change their knowledge of that. So the last thing I wanted to talk about experimentally—
You have 1 minute, Adam.
Okay, thanks, I'm almost done. Was a question of how that neuron sort of gains that innate knowledge. It could be based on its position along the AP axis, or we thought maybe it could be based on its memory of where it has innervated. And we knew this really interesting situation where when we did this crizotinib treatment, we sort of dissociated the position of these neurons from their target. If you remember, these anterior neurons that would normally go to 4 were instead going to PA6. And so if we transplanted them, we could ask, do they go based on their history to PA6 or based on their position to PA4? And I'll just quickly say that the answer is they go based on their position. So an anterior transplanted neuron that would in its original state have gone to PA6 and during development now is going back to PA4. So it seems like it's not history, it's not memory of where it was, it's just its position along the AP axis. And so just to quickly conclude, we've identified that there are these really different mechanisms at play between development and regeneration in this case, where in development, you've got this cell extrinsic temporal patterning mechanism regulated by retinoic acid and MET. And then during regeneration, we seem to have this cell intrinsic spatial patterning mechanism driving these cells back to the right targets. And I didn't have time to show the data, but we actually know now that retinoic acid and MET have nothing to do with this. And so just to give you a quick idea of where we're going with this regeneration project, we're clearly kind of mid-project here. We're really interested in knowing what are the molecular determinants of this intrinsic knowledge of where to send an axon. And so we found that if we photoconvert individual branches here, we can use that to label all of the cells that innervate each branch. And so we've done that for all the branches, and then we're isolating and sequencing these cells to understand what are the gene expression differences between each target group that might give us an idea of the molecules that are determining axon targeting during regeneration. So with that, I'll thank the lab, especially Cecilia, Jason, and former grad student Gabby, who were really important contributors to this project, and I'm happy to take questions.
Great, thank you, Adam. That was really nice. Okay, so we will start with Jennifer Mitchell asks, do you know if this wave of retinoic acid affects other tissues and cells in a similar pattern from posterior to anterior?
That's a good question. I've not looked at anything other than this. So this is happening probably in the retinoic acid wave, like 30 to 40 hour post-fertilization time range. So it's sort of, a lot of development has already happened, but I haven't looked at any other neural tissues to see if there's other things that are being regulated by that wave, that receding retinoic acid.
Okay, Bruce Ostro asks, or says, a classic example of neural specification comes from a chick study, whereby if the developing neural crest from thoracic segment 7 through lumbar segments 3 are reversed experimentally, such that L3 is now anterior to T7, then those neurons still innervate their normal target, such as T7 to the sartorius muscle in both control and reversed embryos. This is different from your AP transplant experiment in zebrafish. How so?
Yeah, so that would suggest that those cells kind of retain an innate knowledge. We've seen that in the eye too, where if you turn the retina around, everything kind of still goes to the right place. And so that would suggest that either they innately know, or whatever environmental signal is involved is sort of also flipped, depending on how much tissue you're flipping. In this case, yeah, I think you'd see the opposite, because if you flip just the neurons themselves, they would still be subject to that retinoic acid gradient, and so they'd all change what they were doing. Hopefully that answers the question.
Okay, Julia Oswald asks, how would you predict neurons that were derived from stem cells in vitro would behave after transplant? Thinking of laboratories that work on cell replacement.
So it's hard to ask a follow-up question, I guess. I'm going to guess that you mean in the regeneration context, because that's when I think about sort of cell replacement, I think more about the regeneration context. Yeah, it's a great question. I don't know the answer to that. We've shown via transplantation that you can take a cell and you can place it back and it'll do the right thing, right? It'll reincorporate, but that cell, has already been specified in vivo as a vagus neuron. So I think that there is a lot of hope that if we could properly specify those cells and then put them in, they'd be able to reincorporate. But I don't know how far along the specification route they would have to go before we're able to insert them.
Okay, Brandon Carpenter says, this is a super cool system. Do the regenerated transplanted neurons function normally?
Yeah, that's a really good question. It's not something that we've looked at yet. There are some, we're working on some tools and we have some ideas to actually test whether they're functionally reintegrating into these circuits. But so far, I don't have an answer for you yet. They look, they look good from a just, axon targeting perspective though.
Deepam Gupta asks, what is the source of the retinoic acid gradient?
So I think probably it is the somites. We know that they make retinoic acid for a while at least. It seems like actually, I think what might be happening is that so the somites kind of fall a little bit posterior to the vagus neurons. And so they would be a good place to provide a sort of posteriorly sourced signal. And what seems like is happening actually is that I think they're turning off the synthesis of retinoic acid fairly early in the process of this nerve developing. And I think that the turning off of retinoic acid synthesis might be part of what's driving that recession over time. That's our best guess, but I haven't verified it.
So this is a question from Alex Nechukarik. Apologies, Alex, for that. He says, great talk. What is the temporal and spatial relationship between cranial, sensory, and motor innervation in the vagal region? Which one extends first? And do you need cranial sensory neurons for correct peripheral innervation?
Yeah, so that varies by cranial nerve. So in the case of the vagus, the motor neurons extend their axons first. And then the sensory, so there are sensory neurons down here in the periphery that extend axons back up. And what's actually been shown for the vagus is that the sensory neurons actually require the motor axons in order to get back. So if you fail to grow out the motor axons, you will also fail to grow the sensory axons back up. And I said that's different. There are more anterior cranial nerves where the opposite is true, where actually motors require sensory. But in this case, sensory requires motor.
Very good. Here's a question from someone you know, Sally Horn-Adonovec. She asks, when you transplant one cell, the axon can track and extend along the existing branch. If you could cut the branch innervating a given arch so that no guiding axons are left, can the axons still find their way? Yeah,
that's a good question. So the answer seems like not. So I've actually done this experiment in reverse, where I take a wild type anterior and I put it into a crizotinib treated animal where now there is no innervation of 4. So I'm trying to get a cell to grow back to 4 in a case where there is no previous innervation of 4. And what we end up seeing is it can't go to 4. It'll go to sort of the anterior most branch that has been innervated in that animal. And so it does seem like these regenerating axons sort of require previous innervation or existing innervation by other axons in order to get where they need to go.
And you have one more question from Deepta Vasudevan. Have you tried ablating, severing the entire axonal bundle to see if the axons can regrow to the right targets?
I have. So this is going to seem a little bit conflicted to what I just told Sally. So we are looking at severing the whole nerve. So the way I've done that is I can transgenically label a small number of cells. I can look at where their axons go developmentally, and then in larvae I can sever the nerve and let it regrow. You get pretty good regrowth of all of the branches. And so far it seems like these cells are actually pretty good at finding their way back. So this is a case where we had some neurons that were going to the visceral branch before and that regrew to the visceral branch after. And so in this case, obviously you don't require pre-existing innervation in the case of whole nerve severing because we've sort of lost all innervation that was there. So we're still trying to work out the differences there, but yeah, we have tried the laser severing approach.
Okay, that is the last question. So with that, I would like to say thank you very much to Melissa and Adam for your very excellent talks today. And they were both very encouraging and well presented. This seminar has been recorded and it'll be available on the SDB website starting next Tuesday, not Monday, since it's President's Day, a holiday. And you can join us in a month on March 12th, which is a Friday, for Jubo New from Harvard Medical School, rather, and Nabora Reyes de Morel from the University of California, San Francisco. Thank you, everybody, and for coming. Please stay safe and have a nice weekend.